Okay, we'll start for chapter number 13, that is lens, its anatomy and metabolism. Okay, let's start from the lens, how the lens actually looks. So, if I may, it is the anterior part of the lens and it is the posterior part of the lens. Okay, so if we draw the equator, one thing is very clear, that it is not same. Okay. So the lens is obviously a biconvex structure, both of the sides are convex, but it is more, it is less. How much more? The posterior anterior radius of curvature, it is more flat, so it is 10 millimeter, and the posterior radius of curvature, that is more convex, the radius is 6 millimeter. Now it looks a little bit weird, but or we can say it 96 or 69, like that. Equator, it has a diameter of 10 millimeter as well, so it is also 10. Remember, the curvature of radius of curvature for a square, the radius of curvature is infinity. So the more flat the surface is, the more radius of curvature it has. Okay, now comes more questions. It is biconvex, it is transparent in color, it is avascular, that means no blood supply. Avascular is equal to no blood supply, just like cornea, same as cornea. Next we have where the position of the lens. Okay, so we know it is lens, obviously we have the entire eye here resides. This part where the lens actually resides, it is called patellar fossa. So this part where the lens actually resides, it is called patellar fossa, but not so important. The refractive power of lens, is 6 centimeters, so it is 16. 10 plus 6 is equal to 16 diopters to 19 diopters, just changes. So refractive power and what is the most refractive power? Yes, there is the previous year question. The most refractive part is the center of the lens. Central part, central lens, most refractive power and maximum refractive index in eye. Obviously lens is the main structure to actually converts the rays. Lens is the main structure, the others are just supporting structures. So what is the refractive index of eye? It is 1.6, or we can say 1.4 actually, so, but we will remember 1.6, it will be easy. 1.4 actually, 1.6 will remember this. Next, where from the lens develops? Surface ectoderm, not so important. Okay, now there is a little bit more anatomy here. If we see, this is my cornea, this is my eye, and here is my optic nerve. Here is other structures. Here is my lens. Here it is the anterior chamber, it is the posterior chamber, vitreous chamber. The space between the vitreous chamber and here we have the zonules, we have the muscles. So space between the vitreous chamber and the lens, it is called Berger space. It's equal to retrodental space. And the attachment between the posterior surface and vitreous, posterior surface of lens and the vitreous, it is called Wegener's ligament. Both of these are never asked before, so I am not actually very much interested in this. Here comes an hyaline artery from the optic nerve to the lens. Okay. It is the hyaloid artery. It dissolves, disappears in the 8th month of gestation. 8th month gestation, dissolved, disappear. Okay. If it doesn't disappear and it remains behind the back of the lens, if it doesn't disappear and remains in the back of the lens, if it remains in a hair, so it is called Mittendorf dot. And if it doesn't disappear in the optic disc, obviously it is the optic disc here. It is the optic disc. If it doesn't disappear from the optic disc, it should be, it's called Bergmeister. So if it remains on the lens, it is called dot, and it remains in the blind spot or optic disc, it is called papilla. Next comes the layers of lens. Let's make a lens from a child. So we have first the embryonic lens. Let's make the nucleus. It is the embryonic lens. Okay, so I can just make this picture here. This is the embryonic lens. This is actually working properly. Okay, it is the embryonic lens here. Embryonic center, it contains primary lens fibers. So here contains is the primary lens fibers. Now it is surrounded by second layer, that is fetal lens. So we have embryonic, now we have fetal lens. Next comes the infantile lens. Infantile lens. It is becoming larger. We have finally the adult lens. After infantile, we directly go to adult lens. All of this part, I can say it is just the nucleus. All of this infantile, all of this part, entirety is the nucleus. Now, what comes next? Next comes is the cortex. About the infantile, we have cortex. It's the cortex. And both nucleus and cortex, they contains lens fibers. This also contains lens fibers. Among the lens fibers, embryonic contains primary and the rest of them contains secondary lens fibers. Okay. Secondary lens fibers. It also contains lens fibers. Next comes the most important part, that is the epithelium layer. Epithelium. The funny part is there is an anterior epithelium, but there is no posterior epithelium. There is no posterior epithelium. Okay. And the most outer part, that is simply the capsule. Capsule surrounds the entirety of the lens. Capsule. Now, important questions regarding all this. Capsule is the thickest basement membrane in the entire body. It is more thick in the anterior pole, but obviously as it approaches the posterior part, it thins, it becomes thin. So capsule, anterior capsule, thickest basement membrane. Basement membrane is made up by collagen type 4, collagen 4. Okay, it is for the entire body. Okay. Next, there is epithelium. This epithelium, it has two type of cells. One is A cells and other one is E cells. A cells does metabolism. And what does the E cells? It does the most important things, that is mitosis. It causes division. Mitosis means new lens is formed by the epithelial layer. This layer forms the new lens. Okay, it forms the new lens fibers actually. Posterior epithelium is absent, so there is only anterior epithelium. There is no posterior epithelium. No posterior epithelium. Now, obviously, it is the one thing means that this is here forms the new layer, new lens. So under this, beneath this cortex, it contains the youngest lens fibers. It contains the youngest lens fibers. Then it becomes old, more old, much more old, very much old. Okay, so from here the new lens comes and it obviously gets inside and inside, pumped out. Then we have the nucleus, we have the four zones, and this is the entirety. There are some sutures in the entire lens. Sutures formed during fetal period. There are two types of sutures. One is anterior suture, Y-shaped, and obviously it is the anterior one. And there is an inverted Y-shaped posterior suture. Okay. Now comes the proteins of the lens. We have two types of proteins. One is water soluble. We have proteins. One is water soluble. So definitely the other one will be water insoluble. Now this has two parts. One is urea soluble but water insoluble. Urea soluble and proteins that are water insoluble, and along with that, okay, water water insoluble, and along with that proteins that are also urea insoluble. Okay, now check this. In water soluble we have the alpha proteins, water solubles are called crystalline. Crystallines. Crystallines. And alpha crystallines, that is the largest, takes the heat shock. Prevent denaturation. That means these are the most available ones. Next comes the beta. They are the বেশিরভাগ one, বেশিরভাগ 55%, and last comes the gamma. Not so. Now comes water insoluble but urea soluble. We have, they make the cytoskeleton proteins, and the distribution leads to cataract formation. Cytoskeletal protein, but it also causes to cataract formation. Next come urea insoluble and water insoluble, both of insoluble, that means entirely insoluble. Urea, water and urea insoluble. They makes transparent, maintains the transparency. Example, aquaporin, Mi26. So these are the proteins. Now there are some high molecular weight proteins. Here some proteins: hemestine, philistine, faecinin, high molecular weight proteins. That is the high molecular weight protein, that is HM1, HM2, HM3, HM4. These are obviously water soluble, these are water insoluble. Most important is HM4 is a question that have come before. HM4 increase in nuclear cataract. This is AIMS question. Now comes the antioxidant mechanism of lens. So we have antioxidants of lens. So first one, we have vitamins E, vitamin E antioxidant. We have vitamin E, we also have vitamin C, we also have catalase, we also have super oxide, dismutase, that means SODs. ion reductase. And low oxygen O2 level, low O2 in lens. So we have these ones. If a vitrectomy is done, that means if vitrectomy is done, then low oxygen level will not maintain, environment will not maintain, can lead to cataract. Here's another question, that is vitamin E and CE. These are antioxidant for lens, but vitamin E is not an antioxidant for lens. Vitamin E is antioxidant but not for lens. Now there is a question comes lens paradox. Writing up here. Lens paradox. Paradox means in old person, in old age, the curvature of lens becomes more. So curvature increase implies power increase, but also the refractive index decrease. That means power decrease. Ultimately it sums up to balance. So it is a balance. Now comes to the metabolism of lens. The metabolism of lens. Glucose is diffused from aqueous mostly. We know we have aqueous humor, and here we have the vitreous humor. So glucose comes from this, mainly from the aqueous humor. So we have the lens. Glucose comes mostly from the aqueous humor. Aqueous humor, less from the vitreous humor. Now this glucose has 3 pathways. Mostly like anaerobic glycolysis, cis, it is mostly, so 80%, mostly actually. Next there is Krebs cycle and path. Next is Krebs cycle or hexose monophosphate shunt or hexose monophosphate shunt, and the most important one is the sorbitol pathway. Sorbitol pathway. In the sorbitol pathway, what happens that glucose through aldose reductase becomes the sorbitol. Aldose reductase glucose becomes sorbitol salt, yes, sugar, sorbitol sugar. It accumulate in lens, osmotic pressure increases, that means it draws more fluid inside, so fluid in lens and causes what? Obviously opaque, it makes it opaque, that means opacification, or in short cataract. So where it happens actually? It happens in two patients, that is diabetes, they have no sugar, and galactosemia. Now there's ascorbic acid. There's another question comes, that is the ascorbic acid transport. Ascorbic acid in lens transported by what? Sodium dependent is PCT2. It is a active transport. I'll find this again. Okay, so this is the entirety of today's class.